Recall the central dogma of molecular biology. The flow of genetic information usually goes from DNA to RNA to protein. And at each step, replication to transcription to translation, there are a series of on and off switches to regulate this. This enables our genetic machinery to determine which genes are expressed, where they're expressed, as in which cells do this, and when they are expressed. That timing could be different in the lifespan of the organism, but also be finely controlled to ensure quantity differences, how much is made at any given point in the day. This level of regulation is important for cellular control and differentiation. Because recall that the genetic content of every somatic cell is the same, but not all genes are expressed in every cell. We don't want our eyes secreting gastric acid. It is the differential gene expression patterns that arise in these different cells that ultimately will give rise to a complete and functioning organism. That genetic control of expression will determine whether a cell becomes an eye or a liver. Due to their simplicity, there's not as much level of control. They have no nucleus. The DNA is freely floating, which means transcription and translation can occur simultaneously. There's nothing to stop it. And so everything there is made on demand. And when stopped, the levels will decrease because they're being utilized. So for prokaryotes, all regulation occurs at the transcriptional level. To help ensure that you're transcribing and translating everything you might need for a specific task, Jacob and Monod in 1962 announced the prokaryotic model of gene control, the operon. They hypothesized that proteins that are needed for a specific function or involved in this same biological pathway would be encoded together, and went on to prove the existence of an operon. These operons consist of a regulatory gene, a promoter, and an operator, followed by the genes of interest for this common function. The regulator may be an inducer and help to turn on the gene, or a repressor and turn off the gene. The promoter is an attachment. The promoter is an attachment sequence on the DNA for RNA polymerase, which will encourage gene expression. The operator is the switch. This is the binding site for the repressor or the inducer to help stabilize polymerase. There are two operons in particular that are considered the classic models for understanding this level of gene expression. The first is the TRP operon. This is a repressible operon, meaning it is turned off when not necessary. Tryptophan is an essential amino acid. It is required that we eat this. Our bodies need it to survive. And so if tryptophan is not present, we have to have a mechanism to make our own tryptophan, lest we run out. But efficiency is the key in biology. So if tryptophan can be freely obtained from the environment, we don't need to spend the resources to make our own. Herein is how a repressible operon works. If the resource, in this case tryptophan, is present, and there's enough tryptophan to activate the repressor will bind to the operator and prevent the polymerase from moving forward to transcribe the structural genes E, D, C, B, and A. However, if tryptophan is being utilized by the body and not present in one's diet, the repressor itself is inactive and cannot bind. So when RNA polymerase binds the promoter region, it is freely able to continue on and transcribe the structural genes to make more tryptophan. A comparison is the lac operon. This is an inducible operon, meaning it is only turned on when the resource in question is available. So if lactose is not present, a repressor is bound to the operator, preventing transcription. You don't need to make something that you are not digesting. But if lactose is present, the repressor binds instead to lactose. Lactose acts as a competitive inhibitor. And now the repressor cannot hold to the operator switch, and so polymerase is free to transcribe the enzymes, LAC-C, Y, and A, which then allow lactose to be digested and utilized. Operons ensure that all related genes are expressed together. This increases the efficiency Only one regulatory protein is required to make all things for that particular function. Eukaryotes have a nucleus, creating a spatial segregation of transcription and translation events. There are lots of regulatory factors that can trigger often sequential changes and steps. And so you'll find that in eukaryotes, there's regulation at the transcription level. When do we make the RNA? Regulation at the RNA processing level, regulation at the translation level, and even then, modifications to those proteins before they are activated. The first level of regulation has to do with packaging and access to the DNA. Recall that a chromosome is composed of chromatin, that is DNA wound around histones. And depending upon the stage of the cell cycle, the chromosome structure might be looser or more compacted. Euchromatin describes loose chromatin. This occurs prior to mitosis, prior to the condensing of DNA. In this case, the DNA is open and accessible for gene expression. But during mitosis, when we are busy moving the chromosomes and dividing the cell, that is not the time to be making materials. Recall that the chromosomes have to condense and pack down. They pack into what is known as heterochromatin, these tight structures. There's no free DNA that's accessible at this time. The package forms are triggered by acetylation or methylation of the DNA sequence. If the histone proteins receive acetyl groups, they uncoil, making the DNA accessible. If instead the histone proteins receive a methyl group, they coil tighter, wrapping themselves in DNA, making the DNA inaccessible, the DNA polymerase and other transcription factors required to read the gene information. Eukaryotes also have post-transcriptional control, meaning that after the mRNA is transcribed, it needs some processing. It needs a 5' cap, a poly A tail, and its introns to be excised before it can be translated. There's some preparation prior to protein synthesis. The 5' cap and poly A tail help ensure longevity within the cell, while removing the introns help in speed and efficiency. It also allows for differential expression through a process known as alternative splicing. The pre-mRNA can be alternatively cut and placed together to create different proteins. This is also known as exon shuffling. In the diagram here, we can see 3 green introns that are non-coding regions of DNA that need to be removed from our pre-mRNA before it can be translated. In scenario 1, it reads genes 1, 2, and 3 together. But in scenario 2, it skips gene 2 and instead adds gene 4. By producing these 2 different mRNAs, it can make 2 different end products without coding for any additional information. Further studies involving exon shuffling revealed 5 models of alternative splicing could take place. A model known as exon skipping, where every other is included. Situations where one or the other, but never both genes are grouped together. Patterns directed by alternative 5' donor sites or alternative 3' acceptor sites. And instances where an intron might be retained. These variations in RNA processing determine which sections will be coded for and allow us to express more proteins than genes we carry. It also creates new exon combinations and introduces variation to the organisms. The go or no-go of translation is partly regulated by the lifespan of the mRNA. The mRNA message may live hours to weeks, depending upon the stability of that protein. The 5' cap will inhibit degradation so long as there's a poly A tail. But recall that every time transcription happens, the polymerase cannot transcribe directly below it. And so every copying, every reading will result in a little degradation at the far end, the poly A tail. And while this will not degrade the actual genetic message, if too much of those A's are shortened, the 5' cap will be removed And this is a precursor for ubiquitin tags that signal for ultimate digestion and recycling. If there are no mRNAs available, translation will not occur. There are other regulatory proteins that can control whether or not translation is taking place. Some will bind to the 5' cap and inhibit attachment of the mRNA to the ribosome, or act as enhancers to signal more frequent attachment to ribosomes. The more they can stabilize the RNA, the more likely translation will occur. After translation, there are post-translational controls, including processing the folding of that protein into its three-dimensional shape, because recall that form dictates function. The sequence of amino acids will form into the secondary structure, alpha helices or beta pleated sheets. These will then fold upon themselves, creating a three-dimensional shape, and then come together with other proteins to accomplish their task. Oftentimes, folding is not enough. These polypeptides must be cleaved by enzymes. This activates the proteins, making them energetically efficient. It allows them to not do their task until they're in the area in which they need to be used. In discussing cell differentiation, it's important to identify that a cell becomes specialized through a series of small changes. We refer to this as potency. Its potential. Undifferentiated cells, such as stem cells, have massive potential. They could become anything. But this is a continuum, because as they move forward through the process of differentiation, they lose some of that potential. Totipotent cells are the most robust cells. They can become anything. They are capable of differentiating into any cell of the organism, including embryonic cells outside of the fetus. Pluripotent cells have now specialized so that they are restricted to any cell of the body. They cannot form the placenta or the umbilical cord. Multipotent cells can differentiate into multiple lines, but a more limited cell type. Oligopotent lines are even more committed, restricted to only a few cell variations. And unipotent stem cells have unlimited replication capacity, but only one cell that they can make time and time again. Cellular differentiation describes the divergence in structure and function of these different cell types. It is achievable because of variations in gene expression. Specialized cells may express only a small fraction of the genes of the organism. As adults, we have very limited stem cell lines left to us. In many tissues, they serve as a sort of internal repair system, dividing essentially without limit and replenishing other cells throughout life. Differentiation occurs extensively in fetal development and will continue into adulthood, although the number of cells left to do this will be impaired with age.